Hi everyone, my name is Caroline Larkin, and today I'll be providing a brief overview on my research on Eastern Equine Encephalitis Virus. So, Eastern Equine Encephalitis Virus is probably something you've never heard of, but it is an alpha virus, meaning that it has a positive sense RNA genome. It's found in the Americas and is typically transmitted to humans through mosquitoes, and it can cause severe disease in humans characterized by fever, meningitis, and of course encephalitis, with a mortality rate of 30 to 70% when neuroinvasive. And if that doesn't convince you how scary this virus can be, well, it's so scary that the Dr. Anthony Fauci himself wrote about it, albeit he got a couple things wrong in his article. um, but moreover, to reemphasize, there are no standardized treatments or cures, so patients rely on supportive care only, and it can be transmitted via aerosols, so there is a threat as a biological weapon. And as a result of that, you can only study it in a BSL-3 laboratory. So given all of these factors, we really need to learn more, but it's really challenging to do so experimentally. So in order to further probe triple EV, I'm going to give you a bit of a primer on replication. There are 6 stages in triple EV replication, which are as follows. You have attachment of a viral particle to the outside of the cell via a receptor. Then the viral particle will enter the cell, typically via endocytosis, where it will uncoat to release the viral genome in the cytoplasm, which will then be replicated using this negative sense template to make more of the positive sense. And then it also hijacks the host ribosomes to make the viral proteins. Once you have everything needed to construct progeny particles, they will assemble and export the cell and go on to infect neighboring cells. So although we understand these general steps or mechanisms, the corresponding dynamics remains remarkably unclear, especially at the intracellular resolution. And given that it requires a BSL-3 laboratory, how are we going to further probe those dynamics and identify therapeutic targets? So the main question that I'm trying to answer with my research is, can a mechanistic model guide selection of therapeutic targets? So here's the picture of my actual model. I know it's quite a lot to look at, but it still contains the same 6 steps as highlighted with the color-coded step numbers. I'm not going to go through every single step of this model, otherwise I'd be here for like 6 hours. So I'm just going to point out a few key features. So one thing that triple EV, as well as other positive sense RNA viruses do is they form these cool replication spherules using the host lipid membranes. And what this does is it helps concentrate the replication machinery, and it serves some other functions. And so we include that in the model as shown by this shaded region. And all you need to know for the rest of this talk is all of the RNA synthesis of the positive sense and the negative sense, as well as the subgenome, which I talk about in the next slide, that all happen. This is where all the action happens. So like I said previously, if you're familiar with coronaviruses, they also have a subgenome, but triple EV has a second open reading frame to synthesize the subgenome RNA. And all I need you to know is the subgenome RNA is used to make the structural proteins like the capsid and spike proteins. And one final model feature is we include the production of both infectious plaque-forming units, or PFUs, and non-infectious virus-like particles. Now, we have a testable hypothesis from the model that the main difference or structural difference between these is the presence of a functional genome. And this is something that we're aiming to experimentally verify in the future. So just to summarize, my model is encoded in the BioNetGen language. It contains 36 model species, 60 rate rules, and 37 parameters. Okay, so now I have this model. How can I actually make it biologically relevant? Well, I need some data. And then I need to estimate the model parameters. So first I need to have a cost function. So luckily right here in Pittsburgh, we have a BSL-3 laboratory and BST-3, which is just out the street. And that is where my collaborator works, and he generated this data set. It's composed of the protein luminescence data, strand-specific PCR data, and lastly, PFU production over time, specific to triple EV. And then, but given how big the model is, there is no way we could have good parameter estimates using just that data set. So I include some other biological constraints based off some other observed characteristics of triple EV replication, but for the scope of the talk, I won't talk about it here. But I'm more than happy to talk about it later. But anyway, now that we have our cost function, we have the model, we utilized preconditioned Monte Carlo, and this will give us a probability distribution of our model parameters. And then when we make model predictions, we can quantify the uncertainty of those predictions. So after calibrating the model, you can see that we're able to capture the structural protein luminescence data over time, the RNA dynamics of the genome shown in blue, and the subgenome, which is shown in green. And then we're also able to capture very well the timing and amplitude of infectious particle production. Now, if we take a closer look at whether the virus is producing infectious particles or these virus-like particles, the model is predicting that at early stages of infection prior to about 4 hours, mostly virus-like particles are being produced, and then at later time points, we're seeing that the infectious particles actually start to catch up in production. Moreover, if we actually plot this ratio, what we can see is, again, early, we see the virus-like particles and a shift to the plaque-forming units. And if you're familiar with virology, one thing they like to do is calculate the overall ratio, not in the scope of dynamics. And so our model is making the prediction that the particle to PFU ratio is roughly 16:1, which is remarkably efficient. Like coronaviruses can have like a particle to PFU ratio of like 10,000. Flu, it's like 100,000:1. So triple EV is like uniquely able to make infectious particles. And this is like in alignment with what we've seen in other alpha viruses. So it's a very good first sign that our model is doing a good job making predictions that weren't fed to it. The next thing I wanted to probe with this model is a unique challenge for positive sense RNA viruses. So unlike DNA viruses or negative sense, the genome has to be used for all different processes. It has to be used for transcription of the negative sense. It has to be used for translation of the proteins. It has to be packaged into progeny genomes, and it can even bind to the negative sense to form a double-stranded intermediate. So the first thing that I took a look at was, where's the genome over time? Is it in these cool little replication spherules, or is it out in the cytoplasm? And what we actually see here is it is surprisingly mostly in the cytoplasm. This makes sense in the early stages of infection as the founding genomes are making their way into the cell and these replication spherules are being formed. And then around like 3.5 hours post-infection, we see like a very sudden shift where most of the genome is in these replication spherules as replication is kicked off. But then once we have that going, we see most of the genome being allocated into the cytoplasm from 5 hours post-infection and beyond. Moreover, if we want to look at like what individual processes the genome is being used for, we can see in early stages of infection, up until about 4 hours post-infection, it's being primarily used for translation of the replication proteins or the RNA-dependent RNA polymerase. And then, again, we're seeing most of it in the green line here being allocated for assembly and release. So the next thing I wanted to look at is to further understand the role of these host ribosomes, because the virus hijacks them to translate everything, right? So previous studies, they have included polysomes in the context of viral replication. And so, as many of you probably know, usually on a single piece of RNA, it's not just one ribosome at a time, they form these polysomal complexes. Well, what I wanted to do with this model is, can I perform polysomal profiling quantitatively through parameter estimation, specifically of this parameter called ribosome distance, which is approximately, on average, how far apart are individual ribosomes on a single strand of RNA in the polysome complex. So we can actually provide numbers to like the polysomal profiling that they can do experimentally. And so the model predictions suggest that ribosomes are very densely packed on triple EV RNA. So it's approximately 52 ribosomes are in a single polysome on the genome, and we see that about 18 ribosomes are packed on the subgenome. And this makes sense because the subgenome is about a third of the length of the genome. Now, if I ask you, which one of these do you think is hoarding all of the host ribosomes, you'd probably guess it's the genome, the positive RNA. Well, you're wrong. So in fact, if we look over time, we can see the different forms of the ribosomes where we have free in purple, and then we have in blue, it bound in complex with the genome, and then red is it's synthesized, it's bound to the subgenomic. And if you can remember, the subgenomic is used to make the structural polyproteins. But the model is very confident in this prediction, as you can see that the subgenome is bound to almost all of the host ribosomes that are available to it. So you would think that if we start messing around with the ribosomal parameters, overall this would lead to like a drastic change in the amount of virus, whether infectious or non-infectious, right? Well, that's not true either. And this like blew my mind for like the past 2 months, and I only recently have the answer, this is the first time I'm presenting it, so bear with me. But we did parameter scans of this parameter defining the polysome composition on the x-axis here, and then on the y-axis, this blue line is indicating the resulting plaque-forming unit production. And this gray distribution here is showing our posterior estimates for ribosome distance. So as you can see, as you greatly vary the polysome composition, you're not seeing a whole lot of virus production being changed. And you see that too with the non-infectious particles. So what is going on? Why is this distribution so tightly constrained if it's not important for the amount of particles being produced? Well, it's actually really important for establishing the relative ratios of the positive sense genome and the negative sense RNA. As you can see here. So if we like look at the two different trajectories and then plot the ratios, we can see that this greatly changes as we make the polysomes more sparse. But why is that the case? Why are ribosomes affecting the amount of RNA? Like, that doesn't make sense a whole lot to me. So what I did is I took a look at the model dynamics, and what I did is I took my original fits and I altered the ribosome distance about 5-fold. So here on the left is, you can see the gray dash line is the original genome trajectory. And then in black is the increased ribosome distance. So what you can see is it causes a delay in the amount of RNA that's being reduced, but you still get roughly the same amount. Now, however, if you look at the negative sense template, you can see that it has the same delay, but then there's more of the negative sense being produced overall, and this is quite surprising. And so that would explain why the polysomes are having more sparser polysomes leads to a delayed but increased bursts of RNA replication, particularly in the negative sense template. Moreover, if we go back to the progeny particle dynamics up here are the dynamics of the plaque-forming units, and on the bottom is the virus-like particles. What we can see here, especially with the plaque-forming units, we see a delay, but then more of the plaque-forming units are being produced. So there's kind of this trade-off in which they kind of cancel each other out with this delay, but more, which is why we see this relative insensitivity. And then we can see something kind of similar, but with virus-like particles, but more of an inverse relationship. So overall, triple EV employs a really tightly regulated strategy. It's very tightly constrained, and it has a fine balance. We cannot mess with it too much, otherwise we'll see drastically different dynamics according to my model. And the take-home messages are the genomes primarily used for packaging, and the host ribosome plays a critical role as the temporal gatekeeper, if you will, with triple EV infection. We're going to do experiments and validate stuff. I know I'm running out of time. And then we want to incorporate the innate immune system in the future. Lastly, I would like to thank my lab, my advisor, my collaborators, my funding sources, and most importantly, my dog, Clover.